All right, so in our this lesson, we want to talk about cholera toxin. So, what is cholera toxin? So, just from the name itself, cholera toxin. So, we're talking about the toxin that causes cholera. Okay, so now this toxin gets to be secreted from a virus. So, cholera, as we know, it is caused by a virus. So, the name of the virus, we call it Vibrio cholera. So, the Vibrio or Vibrio cholera, that is the name of the virus that causes what? Cholera. So this disease that we are able to see, the name cholera, it is very deadly. So now here I want to talk about the mode of action of the toxin that gets to be secreted by the what? By the Vibrio cholera. Okay, so this is what we're able to see here. So this is more like the inside part of a cell. So we have got the cell membrane here. So you can see that these blue parts, they are the ones which are hydrophilic heads, and then the black part here, these are the hydrophilic or hydrophobic heads. Okay, so the blue part hydrophilic, the black part hydrophobic. Okay, so now here, so this is the drawing of the virus. So it looks like this. It's a comma shaped virus. Yeah, it looks like a comma, and it has got a what? A flagellum. It has got a flagellum that helps it to do what? To propel or to move easily. Yeah. So now this cholera toxin gets to affect the small intestine of the body. Yeah. So they are going to, as you eat that contaminated food, they are going to move until they reach the what? The small intestine. Specifically the epithelial walls of the small intestine. Yeah. So in short, as I'm talking about this part of the cell, just know that I'm talking about the small intestine. So this is more like the inside part of the small intestine. The cells which are found there in the small intestine. Okay, so now here we want to talk about the what? The mode of action. So let us assume, not even assume, let us take that this guy, the Vibrio cholera, secretes a toxin. So that toxin, it has got what we call domains. So it has got A domain and B domain or subunits. So when I say a domain, I'm just talking about the area. Yeah? So I'm talking about the A area or A subunit and the B subunit. So it has got the A subunit and the B subunit. So the B subunit helps the A subunit to do what? To move. So in short, the A subunit here is more like a boss. So the A subunit, it is the one that carries the toxin part that will affect that body part or body cell so that that person gets what? Cholera. Okay, so the A subunit will be carried. In fact, this guy, since it is deadly, let me even use the red ink here. So like that. Okay, A subunit carries what? Toxin. It is the toxic part of the subunit or the domains that the toxin has. Okay, so here it looks like this. We have got the A subunit on top here, and then down you are going to have what? The B subunit. So the B subunit is just helping the A subunit to do what? To be moved easily. And then this A subunit, it is further again divided into what? Into two units or two domains. I've got A1 and A2. So the A2, it will be helping the A1 to be what? To be moving again. So in short, the A1 now, it is the one that is carrying the real toxic part of the what? Of the toxin that we're talking about here. Okay. So here, this guy will be further divided. It is further divided into two domains. We have got this is A2. Now it is carrying the boss, the A1. A1, the most deadly toxin part, the A1. So this is how it is. So this is, I've said this is what? The B subunit. So the B subunit is carrying what? The whole lot of A domain or A subunit. And again, the A subunit again, it is further divided. We have got A2, which is carrying the boss, the most toxic part of the domain, which is the A1. Okay, so this B subunit will be helping this A domain. So this is A domain. Will be helping the A domain to do what? To bind to the G protein. So in short, this guy is going to bind to the G protein by the help of B subunit. So the toxin that will be secreted by the Vibrio cholera will be binded to the G protein by the help of B subunit. Okay. So now let us talk about, so this is the G protein. Okay, G protein. So now let us talk about, we dive into it, the real action of the toxin. Okay, so now this guy has been secreted by that. So this guy is secreting what? He's secreting the toxin. So that is the toxin. Okay, it has secreted this toxin. So this toxin will move now. They want to enter now the epithelial cells of the what? Of the small intestine that is now in your stomach. Okay, so now what will happen is when they reach this part where there is a cell membrane, they will attach to a protein which we call GM1. So the GM1 protein stands for ganglioside monocytated. So let me even write it here. So this is ganglioside, okay, monocytated. So mono, IA, then Y like that, cytated. Okay, then 1. Why 1? Because we've got a lot of them. We have GM1, GM2, GM, exactly like that. So this is ganglioside monocytated. Okay, so that there is the G protein that is where found at the membrane of the epithelial cells. So that is where now these guys will target. So the toxin that will be secreted, they are going to target the GM1. That is where they're going to bind. After they bind, now the cell itself won't even notice that this is a what? This is a virus. What it will do is it will engulf. In short, what will happen here is endocytosis. So endocytosis, it is the what? Eating part of a cell. Like the cell is engulfing something. So now here, to be specific, this is phagocytosis, which is happening here. So it will be carried inside. Okay, so after they have been carried, meaning here, have you seen what is happening? They have been carried in the form of what? A vesicle. So this vesicle has just been made by a cell. Okay. So in short, this outer part is from the what? From the cell membrane itself. Okay. So in short, endocytosis has taken place, and then these guys now they are inside the cell. They now want to do what? To modify the G protein. They are targeting the GS protein. Okay. The G protein that we talked about, it is GM1. Now these guys, when they are inside, they want to target the GS protein. So the GS protein, that is the one that they are going to do what? To target inside the cell. Okay. So what will happen is these guys will move as they are inside the what? Inside the vesicle. Now when they reach here, so this is the endoplasmic reticulum. When they reach here now, they will start performing as if it is the home ground, like it is their territory. What they will do is they will like try to change their coats, the outer coating. They will try to steal the membrane of the what? Endoplasmic reticulum and then put on it. Like they will try to do many things. They will try to manipulate themselves. So what they do is they will cleave. Yeah, by what we call protease. So protease will cleave this guy, and then the A1 now, the boss, which is carrying the real toxin, which is the most toxic part of the what? Of the A domain. It will now remain here and move out. So as you can see here, we have got an opening here. So the remain there, that is where this guy, the A1 will come out from the endoplasmic reticulum. Okay, so now what is happening inside here? These guys, they are even now trying to search, like they want to make this guy as their home. They are even like looking for the good place inside the endoplasmic reticulum where now they are going to be multiplying from. So in short, they are making this guy as their home. They are making this guy to be what? Their home, the endoplasmic reticulum. They will try to change so that when they come out, they look new. They are new. So think of endoplasmic reticulum as the what? As maybe let's talk about the dressing room. So this is more like the dressing room of the toxin. So this toxin will be taking this as the dressing room. So they are going to be changing everything so that when they come out, they will come with full force. They will now attack and go and modify the GS protein. So this is what is happening here. So now they'll even try to make a home inside there so that they'll be multiplying from there. Okay. So now when they come out, these guys, when they come out, let me even be using a red ink to denote that So this red part here, it is not the toxic part, that it is the nucleus. So this is the nucleus. Let me even indicate here. So that is a nucleus. So now when this guy they come out, let's say the A1 has now come out. So when it comes out, what will happen is, so that is the A1. It is now moving, going to target the what? The GS protein. So now the GS protein, it is connected to what? To what? To three subunits. So in short, this GS protein, we even call it hetero. So this GS protein, where this guy will go and bind, it is called heterotrimeric. So heterotrimeric, what it means is we have got the G protein, which is having three different subunits. So heterotrimeric, okay, heterotrimeric G protein. That is where they're going to bind. So now the hetero part means just they are different. Now the trimeric means what? They are three. So it means this G protein is having three subunits: the alpha, beta, and gamma. As you can see, this green part is alpha, then beta, then gamma. This one. Okay. So in short, these guys where they are seated there is a what? G protein. Okay. So in short, they are connected to the G protein. So now in the inactive form, so this is inactive. It is inactive. Why? Because it is connected to the what? GDP. So this GDP, when, so this is the GDP, what it means is guanosine. So that is guanosine. Okay, that is guanosine, then diphosphate. Guanosine diphosphate. Okay, so that is guanosine diphosphate. So now what will happen is when this guy is activated, what will happen is it will cleave this part, the GDP will come off, and then inside the cytoplasm of this cell, there are a lot of GTPs, and then it will bind here. So in short, the GTP will come and bind where the GTP was. So the GTP, this GDP will cleave, and then the GTP will bind. Now that is what? The active form of the G protein. So in the normal functioning of the body, this protein that we're talking about here, the G protein, the heterotrimeric G protein, it only gets to be turned on by the what? By the help of some neurotransmitters. So inside the body, we've got a lot of neurotransmitters. They are just signals. That signals the what? The body. So when that neurotransmitter binds on the receptor of this G protein, what will happen is they are going to activate the G protein. But it is different when this virus enters through the GM1. So when it enters through the GM1, what will happen is it will activate the this GS protein that we're talking about here by itself. After it is activated, it will even be what? This guy will even fail to be turned off by its own self. So this is what is happening here. So now, this protein that we are able to see here, the GS protein, what will happen is, so this guy, when it binds here, so the reason why this guy, okay, so this guy, the A1, it will come and bind to the alpha. So it is just targeting the alpha. So it will come and bind to the alpha receptor, to the alpha parts. It will come and bind here. And then after it has binded there, what will happen is this guy will do what? Will split. The beta and the gamma will go away, and then the alpha will be moving alone. So it will be moving here alone, and then go and activate an enzyme which we call adenine enzyme. So adenine cyclase enzyme, that is an enzyme that will now be making or transforming ATP to CAMP, or cyclic adenosine monophosphate. So it is activating the adenine. So let me even write it here. So it is activating this enzyme, the adenine, adenine, like that, cyclase adenine. Okay, cyclic adenine. That is an enzyme. So this enzyme, which is in green, this is the enzyme that it will activate. What will activate this enzyme? The alpha. How? Because it has been activated in the active form. This guy will be cleaved from this guy, from the heteromeric G protein, and then to be moving along and then come and bind here, so that cyclic adenine enzyme, which is this one, which is green, will be what? Activated. Now when this guy is activated, how it works is it will be converting ATP, adenosine triphosphate, and converting ATP to what? Cyclic AMP, which just stands for cyclic adenosine monophosphate. Okay, so this is what is happening. It will be converting ATP to what? To cyclic adenosine monophosphate. So, meaning the alpha part has been affected by this guy, the A1, the toxin part. Okay, I hope you are all following. So, this is what is happening here. So, now this guy, what will make that the alpha part to cleave and not to go back and bind again, like to this protein, the G protein, what will happen is this guy, there's a process which we call ADP. So it is called like this. We have got ADP plus the ribosome. Okay, so the moment this toxin part, the toxin, the moment it enters here, before it even binds to this alpha, what it will do is it will steal the ADP and the ribosome from the what? From, let me even use a different color, from the NAD molecule, NAD plus molecule. It will steal ADP and ribosome and bind these guys to where? To the alpha part. So the reason why this toxin, the A1, is stealing the ADP and the ribosome and giving it to this is to make sure that it locks, it to make sure that this alpha won't bind again to the what? To the beta and the gamma. This is what is happening. So this guy, the ADP and the ribosome, these guys, they are the ones that will make this alpha not to come off from this enzyme, the adenylate cyclic. Okay, so this is what is happening. The ADP is more like a lock. It will make sure that it locks the alpha. It will try to modify the alpha subunit here. It will try to modify it, and then it will fail to come off from the adenylate cyclase. Okay, so meaning this guy, the alpha part will come and bind here. The alpha part will come and bind here. Mind you, it is coming here with a GTP. So meaning this GDP, because now it has been activated. So meaning here we can even wrap here. So meaning so that to show that it has been activated, GTP. Okay, so that is the GTP. Now it has binded to where? To this guy. Have you seen this in red? It is the alpha. This is the A1 now. A1 has binded to the alpha subunit with a GTP. The GTP is coming from the what? Cytoplasm. And then this alpha, the A1 of the toxin, this toxin, the A1 which has binded to the alpha, it is adding what? ADP and ribosome to the alpha. So that when this guy cleaves off, it will make sure that it doesn't bind again to this guy. So in the normal setup, what happens is this guy, when it has been activated, the G protein, when it has been activated, it will come off, the alpha will come off, and it also binds to the what? To the same enzyme, cyclic adenylate enzyme. It will come and bind here, and then it will still, the same process will be happening, but the ATP will be converted to the cyclic AMP, but in moderate. And then this alpha part has got what we call GTP activities. GTPase activities. Pase because it's a what? An enzyme. So this is GTPase activities. What it means is the alpha part, this alpha part of the subunit, it is able to do what? To hydrolyze the, okay, let me just use this one. It is able to hydrolyze the GTP to GDP. Okay, so this alpha subunit, it is able to hydrolyze the GTP to GDP. So this is what is being meant by GTPase activities. The alpha has got the GTPase activities. What GTPase activities means is the alpha is able to hydrolyze the GTP to GTP. In short, when this guy GTP has been hydrolyzed to GTP, it means this guy is what? Is off again. because when it is off, it had a GDP. So meaning alpha is more like a switch. When it hydrolyzes the GDP to GDP, it means that the alpha part is off. The enzyme is off and everything is brought here, and they have now attached just like that, like the way they were at first. So this is what is happening here. The alpha has got what? The GDPase activities. But when this guy, the toxin, comes, upon adding the ADP and the ribosome to the alpha, the GDPase activities will fail to work. So it will make sure that this guy doesn't work. So in short, this guy, the alpha, will fail to hydrolyze GDP to GDP. This is what is happening. This is the toxin working. It will make sure that it alters the what? It alters the alpha. So that, because the alpha, it is the switch, it will make sure that that switch doesn't work. Okay, and it doesn't work. If it doesn't work, meaning we'll be producing what? More of cyclic AMP here. Because the alpha is coming to bind to the what? Cyclic adenine enzyme, which is responsible for changing ATP to cyclic AMP. Now imagine if this guy, the alpha subunit, is binded here to the adenine cyclic enzyme, which is responsible, this adenine cyclic enzyme is responsible for transforming ATP to what? CAMP. So meaning if this guy won't come off from this part, because of what? Because of this toxin. Because the switch has been altered, damaged by the toxin. So meaning it won't come off. So meaning more of ATP will be converted to CAMP. And a lot of CAMP will be in the what? Will be in this cell. So cyclic AMP, like there, cyclic AMP, a lot of them will be produced. ATP will be being converted to the what? Cyclic AMP. Now, a lot of cyclic AMP, when they are a lot inside the cell, what will happen is they are going to stimulate an enzyme which we call protein kinase A. So the A is denoting the adenosine. So these guys which are being produced, being made by the what? Cyclic adenylate enzyme, will stimulate this enzyme. So this enzyme, what you're able to see here, this is an enzyme. This part, that is an enzyme. So this enzyme that you are able to see here, it has got two sides. The regulatory side, that is the regulatory what? Regulatory side, and this active side. Okay, so what will be happening when this guy is stimulated? The protein kinase A, that is the protein kinase A. Let me just draw it here. It is like this. This is the protein. It is a protein kinase A. It is for what? Adenosine. Okay. So like that. So now this protein kinase A, what it does is it will be opening a lot of channels. It is responsible for stimulating and activating a lot of what? Channels. The number one channel that it will open is CFTR. So the CFTR channel will open. The CFTR channel, I've drawn it, it is here. It is here. Okay, this in black. It is the what? CFTR. Now, what it means is cystic fibrosis transmembrane conductance regulator. It is cystic fibrosis transmembrane conductance regulator. So it means this guy is the one which regulates. Okay, so in short, this guy is responsible for what? For taking chloride ions out of the cell. Chloride ions will be going out. A lot of chloride ions will be going where? Out. So they will be leaving the cell. The chloride ions will be leaving the cell. A lot of chloride ions are going to go out like that. Now, take note. When chlorine is leaving, even what will be leaving? Even water will be leaving the cell. So the water will be thinking that let me at least try to balance up things here, because a lot of chlorine have gone here, have gone outside of the cell. Let me try to balance. In the essence of balancing, be happening is more water will be leaving because this guy is still on. It won't be turned off. Why? Because of ADP ribosylation. So this is the ADP ribosylation reaction. We are adding ADP and ribose to the what? To the alpha subunit of the GS protein. So meaning this guy won't be turned off, meaning more ATP will be converted to CAMP, cyclic adenosine monophosphate. A lot of ATP and a lot of cyclic adenosine monophosphate in the cell, they will stimulate which protein? Protein kinase A. What is the function of protein kinase A? Protein kinase A is responsible, it is more like a regulatory protein. So this guy will make sure that a lot of channels open. Number one channel is what? CFTR here, which is there. What is the function of CFTR? CFTR allows chlorine to go out of the cell. So meaning, as the chlorine is going out, even water will be following. And when water is following, it will cause what? Extensive, let me just say diarrhea, severe diarrhea. So meaning severe diarrhea will be caused. Why? A lot of water is leaving the cell. This is what is causing cholera, or this is what this guy, the toxin, does to the cell whenever it enters the epithelial cells of the what? Small intestine.